Thank you, Dr. Snyder. We will now hear from Dr. Linda Buck, who has been a member of the Neuroscience Prize Advisory Board for the past 4 years. Dr. Buck is an investigator of the Howard Hughes Medical Institute, member of Basic Sciences at Fred Hutchinson Cancer Research Center, and affiliate professor of Physiology and Biophysics at the University of Washington. Dr. Buck's research has provided key insights into the mechanisms that underlie the sense of smell in mammals. Her laboratory is currently investigating the mechanisms underlying olfactory perception and the determinants of aging and lifespan. She received a BS from the University of Washington and a PhD from UT Southwestern Medical Center in Dallas, and then conducted postdoctoral research at Columbia University. She was previously a professor of neurobiology at Harvard Medical School. Dr. Buck is a fellow of the American Association for the Advancement of Science and a member of the National Academy of Sciences. She's been the recipient of numerous honors and awards, including the Lewis S. Rosenstiel Award for Distinguished Work in Medical Research, the Gairdner Foundation International Award, and in 2004, the Nobel Prize in Physiology or Medicine. Please welcome Dr. Linda Buck.
Thank you for that very nice introduction. I'd like to begin by thanking the Peter and Patricia Gruber Foundation for the wonderful support that they've given to scientific research and also to human rights. And I'm honored to be here today to celebrate the Gruber Foundation and its contributions. So we're interested in the mechanisms that underlie perception and instinctive behaviors in mammals. Through the sense of smell, humans and other mammals can perceive an enormous number and variety of chemicals in the external world. The olfactory system, which controls the sense of smell, is characterized by exquisite sensitivity and discriminatory power. Even a slight change in the structure of one of these odorants, so-called odorants, can alter its perceived odor. For example, these two molecules are similar in structure, and yet one smells like rose and the other one like sweat. In addition to odorants, the olfactory system also detects pheromones and predator odors that can elicit an instinctive behavior, such as aggression or fear. Now, over the years, we've been especially interested in two questions. How does the olfactory system detect so many chemicals in the external world? And second, how does the nervous system translate environmental chemicals into perceptions and behaviors? Most mammals have two olfactory sense organs, the olfactory epithelium of the nose, or OE, which detects odorants, and the vomeronasal organ, or VNO, the base of the nasal septum, which is thought to be specialized to detect pheromones. Signals from these two olfactory sense organs travel through different neural pathways in the brain. The odor signals generated by sensory neurons in the OE are relayed through the olfactory bulb to the olfactory cortex and then sent to numerous other brain areas involved in perceptions, emotions, and physiological responses. In contrast, signals generated in neurons in the VNO are relayed through the accessory bulb to the medial amygdala and then sent to the hypothalamus, an area that controls virtually all instinctive behaviors and basic drives. Now, in the 1990s, we and others identified 3 different families of chemosensory receptors in the olfactory system. One large family with about 1,000 members in the mouse of odor receptors, or ORs, which Richard Axelrod identified in 1991, and 2 smaller families of receptors that we and others identified in later studies in the VNO. All 3 families are members of the G protein coupled receptor superfamily, receptors that have 7 transmembrane domains, and members of each of these families are extremely variable in amino acid sequence, suggesting that each family
is likely to be able to recognize a large variety of different chemicals. Now, the large size and diversity of the OR family explained how it is that we can detect so many different odorants with different structures. The discovery of the multi-gene family coding ORs also did something else of importance. It provided a set of molecular tools that could be used to explore how environmental chemicals are translated by the nervous system into odor perceptions. Now, in the olfactory system, the olfactory sensory neurons in the OE each send one axon to the olfactory bulb of the brain, where the axon synapses with the dendrites of bulb mitral cells and other bulb neurons in a spherical structure called a glomerulus. Each mouse bulb has about 2,000 glomeruli arrayed over its surface. Each glomerulus receives input from 1,000 to 3,000 sensory neurons. And in the glomerulus, those sensory neurons transfer information to about 25 mitral cells, which then send information to the olfactory cortex. Now, early on, Richard Axel's lab and my lab, working in parallel, asked how it is that inputs from 1,000 different receptors are organized in the nose and then in the olfactory bulb to ultimately generate different odor perceptions. These studies showed that each neuron in the OE expresses only one allele of a single OR gene out of the 1,000 genes. Each gene is expressed in a small subset of neurons that are scattered in an apparently random fashion throughout one of 4 different spatial zones in the nose that express different sets of OR genes. So these studies indicated that the OE is actually a mosaic of neurons expressing different ORs, each with only one type of OR, and those neurons with different ORs are interspersed. There's no odor spot in the nose, which was something that was previously proposed. There simply could not be. Now, in the VNO, the arrangement is very similar, but here we see the V1Rs and V2Rs expressed in 2 different zones with different G proteins, GI2 versus GO. When we looked in the olfactory bulb, we saw something very different from what we had seen in the nose. Here, single OR probes, gene probes, labeled odor receptor messenger RNA in sensory axons in only 1 to 2 glomeruli at 2 spots, one on either side of the olfactory bulb. We found that different OR probes labeled different glomeruli, and that those glomeruli had nearly identical locations in different individuals. So these findings indicate that information provided by 1,000 different ORs is first broadly organized into 4 large sets in the nose, and then is transformed in the olfactory bulb into a highly organized and almost stereotyped sensory map. Despite the difference in the organization of OR inputs in the nose and the bulb, the two have one important thing in common. At both sites, inputs from different ORs are segregated. So each neuron in the nose and each glomerulus and mitral cell in the bulb is dedicated to only one type of OR. So the information is being organized in a series of 1,000 different channels, each carrying input from only one type of OR. The stereotype maps in these bulbs are the same, more or less the same in different individuals, and they're bilaterally symmetrical in the 2 bulbs of the same individual, and in fact each bulb has a mirror image map on its medial and lateral side. So how do 1,000 different ORs encode the unique identities of a multitude of different odorants. To explore this question, Bettina Melnick and our lab used a combination of calcium imaging of single dissociated neurons and single-cell RT-PCR. What she found was that one odorant is recognized by multiple receptors, and one receptor can recognize multiple odorants. But importantly, different odorants are recognized by different combinations of receptors. This indicated that the odor family is used in a combinatorial fashion to encode odor identities. Different odorants are recognized and thereby encoded by different combinations of receptors, but each receptor serves as one component of the codes for many different odorants. Even if each odorant were detected by only 3 ORs, this combinatorial coding scheme could, at least in theory, generate almost 1 billion different odor codes. Now, these studies also provided some interesting insights into some curious features of human odor perception. As I mentioned earlier, a slight change in the structure of an odorant can alter its perceived odor. We found that odorants with very similar structures but different smells invariably had different receptor codes. So if you change the structure of an odorant, even slightly, you can change its code, its receptor code, and that can lead to a change in perceived odor. Altering the concentration of an odorant also changed its receptor code. By increasing the concentration, new receptors were recruited into the odor response. This may explain how it is that changing the concentration of an odorant can change its perceived odor, something that can happen with many odorants. So together, the experiments I've discussed so far indicated that the code for an odor in the nose is a dispersed ensemble of neurons each expressing one OR component of the odorant's receptor code, while in the bulb, the code is a specific combination of glomeruli that receive input from those particular ORs and whose spatial arrangement is essentially the same in different individuals. So now I'd like to turn to a different question. How do pheromones elicit instinctive behaviors and hormonal changes in animals? To explore this question, we decided to focus on GnRH neurons, a small subset of neurons in the hypothalamus that are master regulators of reproduction in mammals. GnRH neurons secrete gonadotropin-releasing hormone, or GnRH, which acts on the pituitary to control reproductive hormones in both males and females. GnRH injection into the brain is also reported to stimulate male and female sexual behaviors in rodents, suggesting that GnRH neurons are also involved in neural circuits that control sexual behaviors. To explore the neural circuits underlying pheromone effects on reproduction, Uli Baum, a postdoc in our lab, prepared transgenic mice in which a transneuronal tracer, BL or biolectin, was expressed in GnRH neurons. The idea was that the GnRH neurons would produce the BL, and it would be transferred across one synapse to label downstream neurons that receive signals from GnRH neurons, as well as upstream neurons that send signals to GnRH neurons. The downstream neurons would have GnRH containing axons in their vicinity, whereas the upstream ones would not, allowing us to predict which of the BL positive cells were downstream and which ones upstream. This slide shows the locations of BL positive neurons in red and GnRH fibers in green and GnRH neurons in blue in different brain areas along the anterior posterior axis of the brain. We found BL positive neurons in a very large number of brain areas, some of which had GnRH axons and some of which did not, allowing us to predict that some areas, those with the green GnRH fibers, were downstream of GnRH neurons, whereas the red ones were probably upstream neurons. So we were actually quite surprised to discover in these experiments that GnRH neurons, which number only about 800 in mice, receive input from about 10,000 neurons in 19 upstream areas. They send information to about 38,000 neurons in 28 downstream areas, and they have bidirectional communication with another 6 areas. So altogether, these 800 neurons are communicating with about 50,000 other neurons in 53 different brain areas. So I should mention that really, under some pressure from me, quantitated the number of neurons in different brain areas in a number of male and female mice, and we were actually surprised to find that both the number and locations of these BL-labeled neurons were very similar in males and females, except for a few sexually dimorphic areas that had more labeled neurons in either males or females. So these studies indicated that GnRH neurons, which are shown here in blue, are communicating with several different brain areas previously linked to sexual behaviors. These include several areas of the hypothalamus in pink, and the medial amygdala in gray here, which receives input derived from the VNO. We were surprised to find that the GnRH neurons, we were also surprised to find BL-labeled neurons in several different regions of the olfactory cortex, which receive input derived from the OE, from the nose, but not the VNO. So this suggested that and some additional experiments, which I'm not discussing today, suggested that the GnRH neurons receive pheromone signals not just from the VNO, but also from the OE. So how is this happening? How are pheromones being detected in the OE? Are they being detected by a special type of odor receptor, a member of the OL family? Or is there some other kind of receptor, a specialized kind of pheromone receptor in the OE that's detecting pheromones? So in thinking about this, we decided to search for other types of GPCRs, G protein-coupled receptors, in the OE, thinking maybe there's another class of chemosensory receptor in the OE. And Steve Lieber is in our lab to do this, first isolated OE neurons by flow cytometry, taking advantage of the endogenous beta-galactosidase activity of the OE neurons. He then made RNA and from the RNA cDNA from the isolated neurons and used that cDNA in quantitative or qPCR reactions with 379 pairs of primers matching G protein coupled receptors not previously implicated in odor, taste, or pheromone detection. He then used in situ hybridization to assess whether the receptors identified in qPCR were actually expressed by neurons in the OE. In these studies, Steve identified a second family of G protein coupled receptors in the OE. These receptors are called trace amine associated receptors, or TARs. TARs are not related to ORs. They're more closely related to receptors for biogenic amines, such as serotonin, and they were previously proposed to possibly recognize what are called trace amines in the brain. Mice have 15 intact TAR genes, humans have 6, and zebrafish have 57. So the tars are evolutionarily conserved from fish to humans. Now, when we use qPCR to look at tar expression in different tissues, what we found was that all of the tars in the mouse, except for TAR1, are expressed in the OE. And they're expressed in the OE at levels similar to two different OR receptors, ORM5 and OREG. We could not detect significant expression of tars in any other tissue, suggesting that tars might be predominantly or exclusively expressed in the OE, although they might be expressed in small subsets of cells in some other tissues. Interestingly, when we searched EST databases, we found a number of zebrafish tar ESTs, and the majority of them were from the fish OE, suggesting that not just in the mouse, but also in the fish, the tars are expressed in the OE. In situ hybridization showed that tars have expression patterns in the OE similar to those of ORs, with each tar expressed in a small subset of neurons that are very much scattered in certain domains in the OE. Double in situ hybridization showed that different tars are expressed in different OE neurons, and we found no evidence that tars are co-expressed in the same neurons as ORs. So this suggested that there are multiple subsets of OE neurons that use different tars rather than ORs to detect chemosensory stimuli. But what stimuli? What do tars recognize? To investigate this question, Steve expressed different tars in HEC293 cells, cell line, and used a cyclic AMP reporter assay to assess tar responses to a variety of different odors with different structures and perceived odor qualities. He tested over 200 different odors, and the only ones for which he found responses were some volatile amines. And in these experiments, he found ligands for 4 different tars expressed in the OE. The different tars recognize their different odorant specificities. Now, remarkably, we found that 3 of the 4 tars for which Steve found ligands recognize compounds present in mouse urine, which is a very rich source of social cues in the animal. One tar recognizes a compound that is elevated in the urine of stressed rodents and stressed humans. Another one, and 2 others recognize compounds that are elevated in male compared to female mouse urine. And one of those was previously proposed to be a pheromone. One study indicated that one of those is a pheromone, a male pheromone, that stimulates or accelerates the onset of puberty in young prepubescent female mice. But another study could not repeat that, although they used a different assay. We found that one tar, mouse tar, tar5, recognizes extremely dilute urine from male mice, but much less so, or not at all, urine from female mice or from prepubescent male mice. So this suggests that mouse tar5 can discern both the gender and sexual status of other mice based on urinary cues. So as I discussed already, the OR family easily accounts for the discrimination of myriad chemicals. Nonetheless, the OE contains the second class of chemosensory receptors, the tars. The tars are evolutionarily conserved from fish to humans and found in both mouse and fish OE, suggesting that they function as olfactory receptors in diverse organisms, perhaps including humans. At least 3 tars recognize compounds in mouse urine, 1 associated with psychological state and 2 with gender, and 1 a reported pheromone. These findings suggest that, first, TARs are likely to serve a function distinct from that of ORs, and second, that this function may be involved in the detection of social cues, such as pheromones. Now, humans and other primates do not have a VNO. So if there are pheromones in higher primates and humans, and this is not yet known, actually, it's likely that they would be detected in the OE. As I mentioned earlier, humans have 6 intact TAR genes. This is also true of rhesus macaque, an old world monkey, closely related in evolution to humans. 6 intact TAR genes, some but not all the same as those found in human. Now, Lisa Horowitz in our lab was interested in TARs in humans. We couldn't obtain intact tissue, OE tissue, from humans, but we were able to get some from some other researchers working on macaques. So she cloned the macaque ORs and some macaque ORs and all of the macaque TAR genes. And what she found was that macaque shows patterns of gene expression similar to those seen in rodents. Using an olfactory marker protein probe, she saw labeling in most or all neurons in the macaque OE, and 2 OR probes labeled neurons that were scattered in the OE, just like what is seen in rodents. Actually, no one has ever done experiments to look at the expression of ORs in higher primates before, so this was something new. So far, Lisa's found evidence that at least 5 of the 6 macaque TARs are expressed in the OE, similar to macaque ORs and TARs and ORs in rodents. Each TAR is expressed in a small subset of neurons that are distributed in the OE, not clustered. So these results strongly suggest that TARs will be found in the human OE as well. And one important question, of course, is what do the human TARs recognize? The most intriguing possibility, of course, is they recognize compounds released from other humans. But that remains to be seen. I know that preparatory to this, but we hadn't gotten approval to do this yet, Lisa brought in some things from home that later we found out she couldn't use. But one person in the lab was surprised when he opened the freezer door and a dirty diaper fell out from one of her kids. But anyway, we don't know anything about that. The experiments were never done. So, okay. So now, having found another family of chemosensory receptors in the OE, we naturally wonder whether there might be additional kinds of receptors in the VNO as well. So Steve went ahead, he did the same thing with VNO neurons that he had with OE neurons to look for other receptors. And indeed, he found a third, very small family of chemosensory receptors in the VNO. Now, these are a little bit different. These are called formopeptide receptors, or FPRs. Humans have 3 of them, mice have 7, and FPRs were already well known because all 3 of the human FPRs and 2 of the 7 mouse FPRs are actually expressed in the immune system. And they're found on phagocytic leukocytes. They recognize N-formulated peptides as well as some non-formulated peptides, and those peptides, the N-formal ones like FMLF, which is made in bacteria, induce chemotaxis of the phagocytes. Now, N-formal peptides are interesting because they're produced by bacteria, mitochondria, and chloroplasts. So it's been proposed, or it's generally thought, that FPRs serve an important role in the innate immune response by attracting phagocytes to sites of infection via the formulated peptides released from bacteria, or tissue damage via perhaps formulated peptides released from mitochondria. Now, when we looked at the expression of the different mouse TARs, what we found was that by qPCR, we found that the 2 immune system FPRs are expressed, FPR and the FPR-RS2 are expressed in a number of different tissues, could be found in cDNA from a number of different tissues, possibly because of blood contaminating all those different tissues. In contrast, the other 5 of the FPR genes look like they were expressed almost exclusively in the VNO, although we could not exclude, of course, the small subsets of cells in some other tissues express these other FPRs. Using in situ hybridization, we found that the FPRs have expression patterns similar to those of the VNO receptors. Each TAR gene, FPR gene, is expressed, again, in the small subset of neurons scattered in the VNO at neuroepithelium. We found that the different FPRs are expressed in different VNO neurons, and again, similar to TARs in the LE, we could find no evidence that FPRs were co-expressed with either V1Rs or V2Rs. So this suggested that there are some VNO neurons that are likely to use FPRs, individual FPRs, to detect chemosensory stimuli. Now, we looked extensively for ligands for FPRs, but we weren't able to find any. But another group led by Ivan Rodriguez in Geneva turned out had concurrently discovered this family of VNO TARs, FPRs, and they reported that some of them at least recognize certain formulated peptides that are recognized by the immune system, FPRs. So they speculated that the role of the FPRs in the VNO is to recognize other mice that are sick on the basis of the formulated peptides released from bacteria infecting the animals. And there's already evidence out there that mice will avoid other mice that are sick, have a bacterial infection. So, and using blast searches to look at the genomes of different animal species, we found that mice and rats each have 7 intact FPR genes. Primates have 2 to 3 each. 1 to 2 are found in dog, cat, and horse, and none, we couldn't find any in cow, sheep, or pig genome sequences. Now, a number of these species are similar to mouse and rat in that they have VNOs. So this suggests that, in fact, the phylogenetic analysis suggests that the FPRs found in the VNO arose fairly recently in evolution in the rodent lineage. And this may, you could speculate that this might endow rodents to actually sense chemosensory stimuli that may not be sensed by other animals. So I'd finally like to acknowledge the wonderful postdocs and students who did the work that I've discussed today. Carrie Ressler and Susan Sullivan did all the studies on OR inputs in the nose and bulb. Anna Burkhardt and Hiroki Matsunami did the work on the VNO. Bettina Melnick, together with our collaborators in Japan, Takagi Sato and Junzo Hirono, discovered the combinatorial receptor codes for odors. Uli Bohm did all the work on generational circuits. Steve Lieberus discovered the TARs and FPRs in the olfactory system. Luis Soraya did the bioinformatics studies on the TARs. And Lisa Horowitz did the work on TARs in the cat. And Lisa and Dawn, together with Steve, did the work on the expression patterns of the FPRs. The phylogenetic analysis of the FPRs was done by David Lieberus and Jessica Lieberus at University of Wyoming. And all this work was supported by Howard Hughes Medical Institute and the NIH NIDCD. Thank you very much.
wondering if the TARS in humans might be responsible for the McClintock effect and all the underarm sweat detection. So that'd be a simple system to look at, identify things like smell.
Yeah, that's an interesting idea. The only real evidence I'm aware of pheromones in humans comes from studies done by Martha McClintock showing that that women who are housed together in dorms tend to synchronize their menstrual cycles. And she did an experiment where she put pads under women's arms and then under their nose and found that would shift the menstrual cycles somewhat. So Snyder is asking whether maybe the TARS might recognize something from underarm sweat. And that's a very interesting question, and we don't know the answer, but that'd be a prime place to look. Yes?
So with overlearning or training for olfactory cues to become, say, aversive in a very strong way, does a pattern expression within a specific odor, for a specific odor, will that change with excessive training or reverse training?
Well, we don't. So Leslie Gage asked whether extensive training or exposure to odors, either as aversive or attractive signals, might change the patterns of expression or the organization of OR inputs in the olfactory system. And no one has really looked at that. It's a very interesting question. But there is some evidence out there to suggest that it may be that exposure to an odorant might help the neurons to survive with a particular receptor that recognizes the odorant. The olfactory sensory neurons are short-lived cells that turn over about every 60 days, and it could be that somehow being stimulated will help them to survive longer. Matt?
Do the axons from the TAR-expressing cells converge and evolve?
So Matt asked whether we see convergence of like axons for TARs like we do for ORs. And yes, we've seen that for two TARs, and we're still working on it because we're kind of interested in where the inputs from the TARs go. And so far for two, we've seen glomeruli that are near each other. We don't know what would be the case for the other ones yet. Yes?
I may have missed it in your talk, but the story used to be simple. You needed the bomber and nasal organ to pheromone detection, and then the main olfactory system did everything else. But that's clearly no longer the case, I guess.
No, and there's other evidence that shows that as well.
So why do we need these two systems in some animals?
Well, I think that the brain is well known for parallel processing. You can lop off part of the brain and a human can still function. So I think this concept has been around for a long time, that you might have redundant functions, and it might be particularly important for something like reproduction, that it's so essential to perpetuation of the species.
So you think there might be redundancy between them rather than serving different forms?
Yeah, it could be. I mean, it could be that they're in part redundant and in part, it could be that they're partially overlapping in function. Thank you.